I'm Elina Zúñiga, and I'm here connected with one of the co-organizers, Dr. Matteo Iannacone, and our fabulous speaker today, Dr. Marion Pepper, that will now be introduced by Matteo. But just one quick reminder before our next global immune speaker is one of our co-organizers, actually, Enrique Veiga Fernandez. So hope you can join again next week to hear to his talk. Thank you.
Thanks, Elina. And on behalf of all the organizers, it is my great pleasure to introduce Dr. Marion Pepper as today's global immunospeaker. Marion earned her PhD in immunology in 2006 from the University of Pennsylvania School of Medicine. And there she worked with Chris Hunter, who also gave Global IMMUNOTALK last year. And in his lab, Marion's work focused on the development of CD4 T cell responses to Toxoplasma gondii. And she continued to study the adaptive immune response during her postdoc with Mark Jenkins at the University of Minnesota. And in Mark Jenkins' lab, she and her colleagues developed cutting-edge techniques to investigate host responses to various pathogens, focusing on the isolation and characterization of the very few endogenous antigen-specific cells. And these studies established new paradigms of CD4 effector and memory T cell differentiation and function, and revealed the early divergence of T effector and T follicular helper cells. In 2011, she was recruited by the Department of Immunology at the University of Washington in Seattle as an assistant professor. And in her lab, she continued to try to understand how to regulate immune cell differentiation such that memory responses against infections can be optimized, whereas those against allergy can be suppressed. And her studies have revealed key differentiation programs and functions of both memory CD4 T cells as well as B cells in response to both pathogens and also allergens. And she created an insectary to provide the means for studying the immune response to the physiological route of Plasmodium infection. In 2017, she was awarded the Borer Wellcome Fund Investigator in the Pathogenesis of Infectious Diseases Award and was promoted to Associate Professor. And she has received many other awards for her outstanding work, and she sits on the editorial boards of Cell Reports, Journal of Immunology, eLife. Marion, we are very grateful that you have accepted our invitation to speak today, and we very much look forward to your talk.
Thank you very much for having me. It's a great pleasure to be here.
Yeah, so as you know, a tradition of the Global IMMUNOTALK is that we ask one question to the speakers to get to know them a little better. So the question we would like to ask you is, what is the single most important advice that you would give to a young scientist that is starting a career in immunology?
Yeah, I thought about this for a long time, and I think in the end, I think what I would advise people to do is to be true to themselves and to do science the way that they love to do science. Whatever it is that got them into this entire endeavor, try to remember that as you go through and stay true to that and stay focused on that.
Yeah, so very true. Thanks, Marion. So without any further ado, I think you can go ahead and share your screen. Perfect.
Great. Okay, well then I will get going. So it's a great pleasure to be here. I'm going to talk today about work that we have done over the last 10 years, really focusing on how allergen-specific CD4 T cells differentiate. I was going to give a subtitle to this, which is why I no longer consider this just a TH2 talk anymore. I used to consider it a TH2 talk, but hopefully you'll see that revealed in the data as I go through it. So let's get it So as many of you know, and as you've heard consistently throughout the pandemic over this year and a half or so, immunity to infection is critical for preventing a secondary infection. And as a virus infects the body, if this red line here is the virus replicating and the blue line here is the immune response mounting to it, hopefully you build an immune response that can control viral replication, and then the immune response can contract again. What's left after that immune response contracts are these cells that we refer to as immune memory, primarily of the adaptive immune response. So B cells, CD4 T cells, and CD8 T cells that have now been endowed with properties that they didn't previously have that enable them to respond quickly and very efficiently to control an infection if they see it again. And what that means is that in response to that same virus or whatever the pathogen is, the immune response that previously took 5 to 7 days to get going and to control the viral replication now can do so in hours. And so I'll show you data later that we can actually get cytokines produced from the CD4 T cell 2 hours if it's a memory cell, whereas it took about 4 or 5 days to get that going if it never seen its antigen before. So my lab is really interested in understanding how these cells form. And specifically, we really focus on how CD4 T cells form. That has led to a large focus of our lab on B cells as well, and B cell memory, because those two cells interact quite frequently. But CD4 T cells are an interesting cell to study because they're super complex. There's no lack of questions to ask with CD4 T cells. One of the reasons is that they're multitaskers. There are many different functional outputs that these T cells can produce. So Th1 cells can control parasites like Toxoplasma gondii by producing cytokines like interferon gamma, while a Th2 cell can produce other types of cytokines that can modulate tissue to clear worm infections. Th17 cells can work to clear fungal infections. And importantly, you can have the same CD4 T cell with the same T cell receptor be activated in a different way so that it can actually turn into any of these different cells. Another thing that CD4 T cells are really important to do is to promote the survival and differentiation of CD8 T cells, both during the priming and the reactivation of the CD8 T cell response. Of course, they're critical mediators of differentiation of B cells. The CD4 T cells, called the T follicular helper cell, migrates to the TB border where it interacts with B cells, and those two cells exchange signals, and I'll talk a fair amount about those cells today, as well as those CD4 T cells that suppress inflammation, called the T regulatory cells, which I will not talk about today at all. So I was really interested in studying all of these different cells and was thinking, oh, maybe I'll study CD4 T cells. And then I went to Chris Hunter's lab and we went to Woods Hole for the immunoparasitology course. And it was there that I also learned that the CD4 T cell also has a better costume, brighter colors, better hat than the B cells or the CD8 T cells. So I really became enamored with this idea of studying CD4 T cells. So that's what my lab has embarked on. We've studied the migratory, phenotypic, and functional characteristics of CD4 T cells. And we've done it in both instances of infection as well as allergen exposure. My love for studying CD4 T cells and infection really came from work that I initiated in Chris Hunter's lab with the Roos lab at the University of Pennsylvania. And it was there that I realized that the development of tools to actually pursue your studies was going to be key to being able to do what I wanted to do. And what I really wanted to do was study antigen-specific T cells. And so in some of the first studies I did in Chris Hunter's lab, we used parasites, Toxoplasma gondii parasites, that expressed the antigen ovalbumin, either in the cytoplasm of the parasite, which you can see here with a stain against ovalbumin, or in the parasitophorus vacuole, which you can see here in between the parasites. And then we could transfer in TCR transgenic T cells that were specific for ovalbumin, called the DO11 T cells. And we had CFSE labeled these cells so that we could track their proliferative response to these different parasites. And in mice that were infected with the parasite where the ova-albumin was retained in the parasite itself, we didn't see distribution of CFSE, which is associated with proliferation. But in those that expressed the ova-albumin into the parasitophorous vacuole and secreted it, we could see the proliferation of these CD4 T cells that had been labeled with CFSE by this distribution of CFSE across the plot. And so this really was eye-opening to me. It was my first glimpse of an antigen-specific T cell response, CD4 T cell response. And I really, I fell for it, hook, line, and sinker. It was the first glimpse of how powerful such a system could be. I then went to the University of Minnesota, where I worked with Mark Jenkins to continue to pursue being able to visualize antigen-specific CD4 T cells and how they respond to pathogens. And there in Mark's lab, working with a close group of collaborators and postdocs and graduate students, we developed a system to generate MHC class 2 tetramers where we could actually look for CD4 T cells that were endogenous and could actually respond to a given peptide MHC complex. One of the first tetramers that I made was this Listeria O tetramer, which allowed us to look for CD4 T cells that could respond to a peptide that was expressed by Listeria. And we could then use an attenuated Listeria model to track how CD4 T cells differentiated, functioned, and were maintained. And what was really exciting for me at this point is that unlike the studies that I did earlier, where we were transferring millions of CFSE labeled TCR transgenic cells and potentially skewing their differentiation, here we actually developed a system where we could find really rare cells so that we could track antigen-specific cells in their native environment using tetramer enrichment strategies. And what this allowed us to do was to find CD4 T cells that were specific for this peptide. This is the LLO peptide tetramer, even in animals that hadn't been infected. We could watch how this emerged, looking at the early response of the infection. So we could see as early as 3 days after infection, the emergence of an expanded CD44 expressing cell population. And that population even grew by day 7, which was really exciting at the time. We ended up being able to track these cells for many, many, many months. And this last time point for this study was 450 days. And it was only through this ability to enrich these rare cells out of the bulk population and develop these very stringent flow cytometry panels to do so that enabled us to track the entire kinetics of the immune response as it proceeded. And this really opened some doors to allow us to look for how different memory subsets were formed. There at the time and still now, although there's more and more memory subsets, I feel like every day, but at the time we were really interested in understanding if CD4 T cells could form the same types of memory cells that CD8 T cells had been described as forming. And there were central memory cells that were lymph node homing and expressed high levels of CCR7. They were thought to be highly proliferative and potentially more plastic. Effector memory cells that were peripheral tissue homing didn't express CCR7 and circulated to the non-lymphoid tissues, potentially having a faster effector function, but less expansion. And then at the time, just recently described tissue resident memory cells, which were thought to be non-residing, non-circulating cells that also didn't express CCR7. So we were really interested in trying to understand, after this Listeria infection, could we differentiate these different populations of T cells in our antigen-specific endogenous populations? And a lot of our thoughts about how to set up these studies and how to think about these studies came from the CD8 T cell field, work from Ananda Goldrath and Sue Keck, John Weary, all sorts of others, really had started to define different transcriptional programs that presented a cell with opposing programs of differentiation. So Transcription factors like BCL6 and BLIMP1 or EMES and TBET, ID3 and ID2, these were associated with different potential for these cells over time. Less differentiated cells were thought to express more BCL6, more differentiated, potentially even terminally differentiated cells express more BLIMP1, etc. So these types of frameworks actually set up the questions that we use to initiate these studies. And this is a little bit complicated here, so hopefully you can see. But one of the first things we did was just to look for our CD4 T cells in the spleens and lymph nodes of mice that had been infected with listeria. This time looking at our LLO specific cells. So these are all gated on tetramer positive cells that we had enriched from the spleen and lymph nodes, and looking for different transcription factors and markers of memory cell populations, both at an early time point, which was day 8, as well as a late time point, which was day 60 at the time. And what we could see, which was really exciting, is that there were actually 3 different populations of cells at this early time point that could be differentiated based on their expression of PD1 and CXCR5, and or their expression of several of these transcription factors. The T follicular helper cell had recently been described. It expressed CXCR5. It was thought to go to the TB border where it would interact with T cells, express BCL6, upregulate BCL6, and help to drive the germinal center. While this other population over here was thought to be more of an effector type of population. But it wasn't clear how these different transcription factors were expressed in these different cells, or if they were retained into the memory phase. And so when I looked at day 8, just to see how these different populations, what their expression of these different transcription factors was, a naive cell, you can see as a naive population of cells is in the gray histogram, but the T effector cells are in black, the TFH cells are in this gray line, and the dotted gray line are the GCTFH cells. We could already see that there were varying expression levels of TBET at this very early time point that was then maintained for these 2 populations at this later time point. We didn't really see the retention of a GCTFH population as the GC had ended by that point, and thought that some of those cells have fallen back into this gate down here. But interestingly, we could also see differences in expression of BCL6 at these early time points, with the TFH and GCTFH expressing more, while the effector cells expressed less. So this really created this interesting paradigm where cells were making this early decision where some of them expressed higher levels of a master transcription factor, while others expressed higher levels of BCL6. And when we looked to see if this related to these populations of memory cells that I just described to you, the central and effector memory cell populations, what was really exciting for us is that we could see that even though both the cell populations that we were looking at expressed some TBET, there was a very different level of TBET expressed by the CCR7 high cells, which were the central memory cells, versus the CCR7 low cells, which are the effector memory cells. And if we also continued to look for these markers of T follicular helper cells, we found that the CCR7 high cells, which were the central memory cells, retained CXCR5, and really that this big population of central memory cells that we were looking at that expressed CCR7 at these late time points also retained characteristics of the TFH cells that we later showed that they were derived from. What was important as well is that we showed that these differences in transcription levels really predicted how these cells were going to respond in a recall response. So here again, just looking at TBET by CXCR5, if we just look at the memory time point, we didn't see any cytokines. This is using intracellular cytokine staining. After injecting mice that had been infected 60 days prior, we then injected them with a peptide, just the LLO peptide, to see if they could respond. So this is what these 2 populations look like. The effector memory cells here, the central memory cells here at rest, just a day 60 time point. If we inject that peptide and then look 2 hours later, we could see differences in the function of these two populations, whereas the effector memory cells that expressed higher levels of TBET also expressed far higher levels of interferon gamma and IL-2 at this 2-hour time point, whereas the central memory cells expressed more IL-2 alone, and then could express some gamma, but not as much as the effector memory cells, keeping with the enhanced TBET expression in these effector cells. So many months, many years later, we put together a lot of information, as did a lot of other labs. A lot of labs worked on this. And there was a burst of discovery around how CD4 T cells make these decisions. And this is a review that we recently published in Annual Reviews from Michael Ruderbush in my lab. And what it shows is that there is this layering of different programs that a CD4 T cell encounters as it gets primed from a dendritic cell. One of the first things that happens when this CD4 T cell gets activated is that it interprets signals from the T cell receptor, from co-stimulatory molecules, that helps to drive this T effector versus TFH decision. The T effector cells will go on to express higher levels of the master transcription factor for that cell, where the TFH cells will express higher levels of BCL6, although they will still retain that signature that they were first gained from this other signal, which is the cytokines that come from the innate cells surrounding the CD4 T cell as it interprets these signals. So in a Th1 response, you then therefore end up with T effector cells that can produce large amounts of gamma and express TBET and BLIMP1, and TFH cells that express less TBET but more BCL6 and are able to express some gamma, which we know is really important for class switching, but not to the same levels that these TFH cells these T effector cells are able to produce. And we can now visualize this framework in real time, and we can use this to test whether this happens early. How does it happen? How does this impact the B cells that these CD4 T cells are interacting with? And just to show some quick preliminary data that I think drives this home, if we do this time an infection with LCMV, we can look for cells that are specific for an LCMV epitope called GP66, again using tetramer enrichment. And what we can see 6 days after infection, again looking at the spleen and lymph nodes, is that we see a population of T effector cells and a population of TFH cells. It's too early for the GC-TFH to have formed. But importantly, if we do this in cytokine reporters, we can track what those cells are making at what time point and how this evolves over time. And so, for example, if we look at IL-4 across these 3 populations, just sort of ignoring the GC-TFH because they're not really there, there's an early little small population of IL-4 producers, but much more robust is the production of interferon gamma from the T effector cells, with far less of it, but still some being produced by these intermediate TFH cells. If we continue to look across time how this evolves, we can see that this population turns into a largely TFH population at day 10, and this can be variable depending on the infection. But now what we can see is there's a little bit more IL-4, especially in these GC-TFH cells that are emerging, and the distribution of interferon gamma is still what it looked like at day 10, with the most interferon gamma coming from the effectors, a small amount of it, but more than it was before from the TFH, and almost none of it, very small amount coming from the GC-TFH. And this was really interesting to us, in keeping with studies from Carola Vinuesa and others showing that class switching generally happens before the germinal center. And we think that these TFH cells may really be mediating that interaction. And you can just see this lastly at day 12. Again, IL-4, now we have a large population of IL-4 producing cells from the GC-TFH cells, but very little interferon gamma. It's now coming down from the effector cells. Those cells may have left already, gone out into the periphery, but very little coming from the TFH or the GC-TFH. And so this framework of differentiation led us to really test what molecules were involved. How does this happen? How do we set up these two different branches of the CD4 T cell response? And we showed in that paper that this early differentiation into the T effectors was dependent upon IL-2, while this differentiation into the TFH cells was really initially driven by TCR signaling and BCL6 upregulation, and later maintained by interactions with B cells, depending on ICOS ligand interactions, cytokines, and other interactions between the T cells and B cells. And importantly, this also led to the production of different memory cells. And so the effector memory cells really came out of this effector response, whereas the central memory cells for a Th1 response really came out of these TFH cells. So that's all well and good for a Th1 response in the spleen and lymph nodes, but I think we all know and accept at this point that for most responses, many of the critical memory cells aren't in the spleen and lymph nodes, but are actually in the tissues. And there are a lot of different programs that are happening in these different subsets of CD4 T cells. So we started to really interrogate how these different programs could emerge, looking both at a different type of T cell, this time the Th2 cell, but also in the tissue where tissue resident memory cells form and can persist. And we decided to do this using a murine model of airway inflammation that recapitulated some of the clinical symptoms of asthma. And asthma is characterized by this acute, intermittent, and recurrent episodes of inflammation. And importantly, it can be induced by a specific allergen, which allowed us to think about how we could create a system where we could actually deliver an allergen and then look for T cells responding to that allergen. The allergen specific CD4 T cells are thought to contribute primarily through a type 2 type of response, although there's a lot of heterogeneity in asthma in people, and there are what people call endotypes, which means very different types of functions of CD4 T cells that can contribute to these allergen-specific responses. And one of the things that attracted us to this question and this problem was this potential to actually look at lung-resident CD4 T cells. And there was a paper that I came across at the time that showed that non-asthmatic recipients of asthmatic lungs developed asthma post-transplantation, while the inverse also happened. Asthmatic patients that received non-asthmatic lungs seemed to resolve their symptoms for at least 3 years. And this really said to us that there was the potential that the asthma was actually being perpetuated in the lungs themselves. And maybe if we could identify the T cells that were involved and what got them there and how to suppress them, we could actually come up with some therapies that would be useful. So one of the ways that we decided to do this was to use the most common source of allergen inducing asthma, which is the house dust mite. And we developed a system where we generated tetramers against an epitope that had been previously described in the house dust mite antigen DERPI1. And we made these tetramers and then very nervously tested them out to see if we could actually find cells that could respond to just an intranasal inoculation of house dust mite allergen. And what we could see with mice that were given 1 large dose of allergen and then 5 smaller doses of allergens is that if we looked in the spleen and lymph nodes, we could use our tetramer to discover these DERPI1 naive cells. There were about 80 of them in the spleen here. But 6 days later, after just 1 bolus of that house dust mite intranasally, we got many thousands of cells in the spleen and lymph nodes, and we could still find those cells, although to a lesser degree later in the response. What was more exciting for us is that when we decided to look in the lungs after injecting an antibody that would label any circulating T cells using anti-Thi 1.2, we could find very rarely some small population of naive T cells that were circulating throughout an unimmunized animal. But if we gave that house dust mite bolus intranasally, we quickly found, we now know it's as soon as 4 to 5 days after inoculation, that T cells that are specific for that DERPI1 specificity go into the lung. Some of these are in the blood, they're still circulating as labeled by the Cy1.2 antibody, while others are actually already going into the lung tissue and are not labeled by the antibody. If we look 3 days after this post-allergic sensitization phase, most of the cells that have expanded are now in the lung parenchyma itself and are not identified by the Cy1.2 label. So this was really exciting to us. We were able to track these cells and show that they persist both in the spleen and lymph nodes for long periods of time, as well as in the lungs. And it gave us a system to start to actually characterize what these cells look like. So again, to avoid issues of cytokines by not catching the right window or looking at transcript instead of protein, which I'll talk about in a second, we used reporter mice that express the human CD2 molecule under the IL-4 promoter or the GFP molecule under the IL-13 promoter to see what types of Th2 cells we were generating, of T cells we were generating. And what we could see in the spleen and lymph nodes that was not surprising based on other people's work is that there were IL-4 cells, IL-4 producing cells that were generated that were specific for our allergen, but they were largely coming from this GCTFH population, which had been described by several labs earlier looking at helminth infections. When we looked for IL-13 in the spleen and lymph nodes, we hardly saw any IL-13 in the spleen and lymph nodes. We also looked for IL-5. We saw same thing there, very little IL-5 or 13 in the spleen and lymph nodes, which is in keeping with the thought that Th2 cells really need to go to the tissue to be terminally differentiated. And a lot of that work was done by Rich Loxley's lab. If we looked in the lung, however, and this time instead of looking at markers of TFH cells, we're now looking at CD69, which can mark tissue resident memory cells. What we could see is the presence of IL-4 producing cells, as well as IL-13 producing cells. And I'll show you some IL-5 data in a little bit. And this was really exciting to us because it suggested that we had caught some window where these cells were being made, where these cytokines were being made, and Th2 cells were truly functioning in these lungs after inoculation with house dust mite allergen. But what was sort of interesting to us is when we pulled out this entire DERPI1 positive population that labeled with our tetramer, only about 30%, 20 to 30% of our cells expressed these Th2 cytokines. And while we were really interested in these cells because we knew that therapeutics that had targeted IL-13 and IL-4 were really effective against allergy and allergic responses, it still really raised the question of what are these other cells that were in here and what are they doing to exacerbate this allergic inflammation if they are. And just to show that we do think that these Th2 cells are important and they do have functions, we showed in the same paper that was published in 2016 that if we knocked out CD4 T cells, we lost the eosinophilia associated with allergic airway inflammation, suggesting the role of IL-5 in driving those eosinophils. We didn't have airway mucus production until we'd instilled the HDM, and we could show that this also, the installation of HDM induced airway hyperresponsiveness, which is associated with IL-13 production. And that again was dependent on class 2 dependent T cells being present. So we knew that CD4 T cells were important. We knew that these symptoms associated with both airway inflammation and a Th2 response were involved, but we wanted to know what other types of cells were in here and how long they lasted and what they looked like. And so we first asked, who are the cells that stick around, and are these really a tissue resident memory pool that can persist for long periods of time? And so we did these studies with Dave Massapust, where Jason Schenkel in his lab would instill the house dust mite allergen into a CD45.1 animal. The mice were conjoined with a congenic partner, and then we could see and look for the ability of the blood to mix and the lymphocytes to mix in both the immunized parabiont and the unimmunized parabiont. When we looked to see if cells that we had basically parked in the lung were migrating out of the lung into the unimmunized parabiont, we could see that if you look in the lung sample here, we could find a significant population of our DRP1 specific cells in the immunized parabiont, but only a very tiny population of cells in our unimmunized parabiont. And if we looked at the phenotypes of these cells that we could recover from the unimmunized parabiont versus the immunized, we could see that the immunized parabiont truly had Thy1.2 negative cells in the lung parenchyma that express CD69, while this unimmunized parabiont, we just happened to catch a few circulating Thy1.2 positive CD69 negative cells going through, and that's just quantified here. So we were really interested in these tissue resident memory cells. We tested whether they could alone induce this airway hyperresponsiveness that we had seen. And to do this, we treated the mice after giving them HDM and the airway inflammation with FTY720, which is an S1P agonist that causes the internalization of the S1P receptor. And what that does, in essence, is trap cells in the lymph node that are in the lymph node, trap cells in the tissues that are in the tissues, and take out the circulating cells out of the equation. And what we could show is that if we did that and then looked to see if airway hyperresponsiveness could still happen, even in the presence of FTY720 with no circulating memory cells contributing to this response, there was very significant airway hyperresponsiveness, suggesting that the lymphocytes that were really retained in this lung are sufficient to drive this airway hyperresponsiveness. So we went on to really look at how these allergen-specific Th2 cells form. We were really curious whether the same paradigm that we had shown for Th1 cells would also apply for Th2 cells. And specifically, were these tissue resident memory cells going to come off of this arm of the effector side of things? As they had a lot of similarities with the effector cells, including low expression of CCR7 and expression of other transcription factors that seemed like they would be appropriate. And so the way that Brian Hondowitz in the lab did these studies is he made mixed bone marrow chimeras between a wild type and a CD25 knockout with mixed congenic bone marrow. Put this bone marrow into a T cell deficient mouse, allowed that to reconstitute, and then perform the airway inflammation system that we'd been using. And then looked for these DRP1 specific cells in both the spleens and lymph nodes or the lungs to see who formed in the absence of IL-2 signaling, as CD25 is the receptor, the high affinity receptor needed for IL-2 signaling. And what we could see in the spleen and lymph nodes is that whether you were looking at wild type or knockout cells, the DRP1 specific cells formed just fine. There were equal ratios in both compartments, suggesting that IL-2 is not required for the proliferation of these cells in the spleen and lymph nodes. However, when we looked at the lungs, we could see that there were very few CD25 knockout cells that made it into the lungs, and this got worse over time, such that if you looked at the memory time point, there were no memory cells that survived from the CD25 knockout compartment. They were all from the wild type. And that was just shown here as a ratio. And we went on to show as well that this also happens in a Th1 response. Brian did these same studies in a viral response, that time looking at intranasal inoculation of LCMV, and showed that, again, IL-2 is really required for these tissue resident memory cells to form, whether it's a Th1 cell or a Th2 cell. So we got to this point where we started to understand how these cells formed. We started to understand how these cells are maintained. I'm not going to talk about that today, but we really wanted to get to how they behave. And most importantly, how do they behave upon allergen re-exposure? So far, we had studied how do the memory cells get there, but we hadn't done very much with how these cells respond to a renewed introduction of the allergen. And one of the first things we wanted to see is, do these cells become quiescent? Do they actually settle down into memory cells? And so one of the ways that we did this, and we've done this several ways now, is to look for the marker of cell cycle Ki67 by CD25 to see if these cells looked like they were not activated anymore, that they were quiescent. And by 30 days after allergic airway inflammation, that whole system had been done, these cells looked like they were resting and were no longer in cell cycle. If we gave antigen at that time point for 2 days, this time giving a small dose of DERPI-1, we could see of HDM, house dust mite extract, we could see the rapid upregulation of Ki67 as well as some cells expressing CD25. And it was possible that these were circulating cells that were coming into the lung over that 2-day time point. So Brian repeated the studies, this time with that same S1P agonist FTY720 on board, and you could see that there were very similar numbers of cells, whether circulating cells could contribute or not, showing decidedly that it was the cells that were actually in the lung parenchyma that were proliferating and expanding. And again, we did this using BRDU and some other tools to further confirm that. So this is a population of cells that is not, quote, terminally differentiated to the point where it can't proliferate anymore. They can be very proliferative, and they can get rapidly activated to respond to an allergen re-inoculation. So we were really curious if the cells that we had described that expressed IL-13 and IL-4 resembled some of the cells that had been described in Th2 responses and allergic responses in humans and in mice as well. And one of the key molecules that had been described as regulating Th2 cells was this IL-33 IL-1 family member receptor, also known as ST2. And several studies had really nicely shown that Th2 cells can upregulate this IL-33 receptor, and that can allow them to respond to IL-33 produced by innate cells or epithelial cells. And one of the studies that showed this was from Nakayama's lab, and you can see here what they did is they in vitro differentiated Th2 cells. And if they looked at those cells in culture after giving them IL-33, you can see that there's very little IL-5, but if you put them into a mouse, you let them form memory cells and then give IL-33 afterwards, take them back out and then give them IL-33, you could see this massive upregulation of IL-5. And so we were really curious if our cells that we had been trying to distinguish as the Th2 pathological cells also expressed IL-33 receptor. And we decided to do this by looking at the Th2 transcription factor, GATA3, as well as the IL-33 receptor. And we looked right after allergen sensitization. We waited and looked at a memory time point, and then we even re-challenged these cells and looked at the, after re-challenge 3 days later. The profile that we could see was sort of in keeping with what we had seen with the IL-13 and IL-4 production, which was somewhere around 20 to 30% of our cells after sensitization expressed, that had expressed IL-13 and IL-4 This was a similar proportion to the frequency of cells that expressed GATA3 and IL-33. This was very different than the profile of the spleen and lymph node cells in the same animal. And again, this is all looking at the DERP1 positive cells across both the spleen and lymph node and the lung. And so we decided that we would really target these IL-33 receptor expressing cells and start to look at all of the other transcription factors that we knew about that could influence CD4 T cell differentiation, and see if these IL-33 receptor expressing cells expressed unique amounts of these transcription factors. So the first thing we did was just to look for our antigen specific cells, which are here in red in the lung, versus our antigen specific cells in the spleen and lymph nodes of the same animals. And one of the first transcription factors that we decided to look at was BLIMP1. And BLIMP1 has been associated with tissue residency as well as effector cell differentiation and function. And what we could see very strikingly is that our antigen-specific cells expressed very, very high levels of BLIMP1, way higher than they do in the spleen and lymph nodes, although there's a small population that still did, and higher than the antigen-specific, non-specific cells, which are shown here in green. We also looked for BCL6. We didn't see very much BCL6 in the lungs of these animals on our DERP1 specific cells, again, using our Thi1.2 labeling. Although we could see it as expected in our spleen and lymph nodes, which marked the TFH cells and the GC TFH cells. We also looked for TCF1, which has also been associated with circulating plastic memory. And what we could see here is that TCF1 was more highly expressed on our DERP1 cells that express CXCR5 in the spleen and lymph nodes, but very low expression in our lungs on our Thi1.2 negative cells that were in the lung parenchyma. So we kept going with different transcription factors. As I said, there are these layers of transcription factors that really give a T cell its function and its identity. So we looked for GATA3, and as you can see from the IL-33 receptor staining that I showed you before, there's the split population of GATA3 on our antigen-specific cells. But we also looked for TBET this time, and we could also find TBET on cells. And a large proportion of cells, about 40 to 50% of our cells were expressing TBET. So this was really exciting to us because it finally was starting to identify who these non-GATA3, non-33 receptor high cells were. We looked for ROR gamma T as well, but we really didn't see ROR gamma T, suggesting that this wasn't really a Th17 response. Now, keep in mind, this is at a memory time point. Earlier on in the response, there is sometimes some IL-17, but at this memory time point, it's not really clear that it's there. So this started to give us some direction as to who these other cells might be, since about half of these DERP1 cells that we were looking at expressed TBET, while the other half expressed GATA3. And it really raised a question of whether there are distinct functional modules within this epitope-specific population that had all migrated to the lung parenchyma of these allergen-treated mice. But I think it's really important just to make the point that cytokines are a little bit tricky. You need to really think about how to look at them, because there are windows of cytokine expression. And even though a lot of people are looking at cytokine production by single cell RNA-seq, a lot of the CD4 T cells can express transcript of a cytokine, but not express the protein itself. And this is just showing this for IL-4. If we use a mouse that it reports basically IL-4 transcript made by Marcus Moores, we can see if we focus on 4 days after immunization in the spleen, IL-4 positive cells, if we gate on our allergen specific cells, if you gate on the naive cells, you don't see those cells. So this is real. And it's pretty equal across both the T effectors and the TFH cells. If, however, we look at that same time point, this time using a mouse that reports the IL-4 protein, and again, this is what Marcus Moore's early work would have predicted, we can see IL-4 production only really coming from these TFH cells and not really being induced yet from these cells that express transcript. So we decided to really embark on this endeavor to understand what cells were making what cytokines at the time that we could watch these different time points. And we started by just re-challenging our allergen-specific cells, looking in different reporters. And we got a lot of help from a lot of people to get a variety of reporters, IL-5 reporters, and we'll go through all of these. But what you can see here is this is looking in the lung, this is looking at our DRP1 positive cells, and this is looking at IL-33 receptor by IL-5. And as predicted by the Nakayama paper, the 33 receptor cells are the predominant IL-5 producing cells in the lung. If we looked at IL-13 production using the IL-13 reporter, we could find that a large portion of the IL-13 producing cells expressed IL-33 receptor, and importantly, all of the IL-33 receptor expressing cells express IL-13, but there are also IL-13 expressing cells that do not express IL-33. For IL-4, we could find IL-4 production from a lot of different cells. It was in different proportions. The 33 receptor positive cells made a large proportion of these IL-4 producers, but so did some of the IL-33 negative cells. IL-2, we could find, this was a reporter from Casey Weaver, we could find IL-2 production from both the IL-33 receptor highs and lows. We could find interferon gamma only coming from the IL-33 receptor low cells, and we've repeated this. And we didn't see very much IL-17, but the IL-17 that we did see in some of these mice, again, came from the IL-33 receptor negative. And so this was really starting to paint a picture for us where we could see that even though these cells had common programs of being tissue resident memory cells, they depended on IL-2 to get there. They made some cytokines in common like IL-4 and IL-2. They still maintained these modules where IL-33 receptor cells expressed more type 2 cytokines and some unique type 2 cytokines. And we have yet to see an IL-33 receptor cell that was expressing interferon gamma. These are very distinct modules here. And the same with IL-33 receptor and IL-17. And we even made double reporters so that we could look at this in the same mouse to see how this was happening. And here you can see the IL-5 and the interferon gamma. And so I think it just really highlights the fact that even though these cells have encountered similar cues to get to the lungs, they undergo, they're responding to the same peptide MHC complex, they've interpreted these differently such that they really have upregulated different functional modules. So these are really Th2-like, these are really Th1-like. We're doing a lot of work right now to dissect the interactions between these two. But I think there's been some recent suggestions that subsetting CD4 T cells and still even relying on Th1 and Th2 subsets is archaic, but I think that that's not true. And I think that there really are these functional subsets that are very much relevant. And I started to think of it, you can tell that I've been in quarantine and doing baking projects because I'm starting to think of it like these rainbow cakes, these rainbow layer cakes. And I think what we're starting to see is that there are many of these layers that are shared between the different subsets that are in the lung that are functioning, but some of the subsets at the top and the overall taste is going to be different because they're unique. And it's this layering of these differentiation states, so becoming a CD4 T cell that can go to the lung and become a tissue resident memory cell and express these localization and adhesion programs, whether it's activated or not, is it rested or is it activated, what kind of cytokines are driving that? But then on top of it, these functional cytokines that seem to be unique to these modules that really all together create the picture of the cell itself and what it's capable of doing. And again, we know that these functional molecules are important because in their absence, you don't get the airway inflammation, or you know, if it's a viral infection, you don't get protection, say if you lack interferon gamma. So we think that these are still relevant. And just one final thought about this is that I think we talk about responses being a Th1 response or a Th2 response, but I think that these responses are far less homogeneous than we really think. And there's remarkable complexity in these responses. And I think through understanding the interactions between these functionally heterogeneous modules, we'll really start to be able to develop some therapeutics that will really start to suppress this response. So with that, I'm going to thank the people who did this work. This was largely done single-handedly by Brian Hondowitz, who has been with me from the beginning. Some of the studies that I showed you early on were from Lila Shahada and Kurt Pruner, who are graduate students in the lab. Former trainees from the lab really got a lot of these studies going. Karen Kim was really key and essential to getting these, and just to thank our funders. And yeah, I'm happy to discuss.
Thanks, Marion, for a fantastic talk and a fresh take on CD4 T cell responses on pathogens and allergens. I hand it over to Elena, who is going to tell you how to ask questions to Marion.
Yes, thank you so much, Marion. That was terrific. So many new insights and bringing up new questions. It was fantastic. And also, I loved the message for the young and not so young scientists. Stay true to yourself. Beautiful. Thank you so much. Okay, so for questions to Marion, search for the account Global IMMUNOTALK, find the tweet that says ask questions for Dr. Marion Pepper here, reply to that tweet with the hashtag Global Immuno, and Marion will answer to the questions using her personal Twitter account, Pepper Marion. So thank you again. It was a fabulous talk, so we appreciate it very much. And hope everyone can join again next week.
Thank you so much.
Thanks, Marion.
Yeah.
Bye.
Bye.
Bye.